Okay. Oops. Okay, so let's start with multiple choice. Okay, first question. Lionfish is a venomous fish found primarily in the Red Sea and the Indian Ocean. In the 1990s, lionfish were accidentally released into the Atlantic Ocean where they found abundant resources and favorable environmental conditions. Which of the following scenarios is most likely to result in the lionfish having a major impact on the communities in which they were introduced? Okay, so again, um, we have a non-native, right, species, right? So when you introduce non-native species, always the chance of becoming invasive, right? Again, probably the most, where's my end? Probably the most likely thing that's going to happen, right, is going to be A, no natural predators, lionfish population will become very large, right? Again, there's abundant resources, right? Favorable environmental conditions, right? Again, resistance to venom doesn't really make sense, right? Random mating would allow them to reach Hardy-Weinberg equilibrium, doesn't make sense. Virus that specifically infects lionfish will become more prevalent. Again, not really sure why the virus would become more prevalent because of lionfish, right? Again, A would be the best and only answer that really makes sense, right? Second one, number 2. Cell communication is critical for the function of both unicellular and multicellular eukaryotes. Which of the following is likely true of cell signaling? Okay, so if we go through the choices, cell signaling uses the highest molecular weight molecules found in living cells. This has nothing to do with cell signaling, right? That is just wrong. Cell signaling has largely been replaced by other cell functions in higher mammals. That is not true, right? Because cell signaling is necessary for your cells to communicate with each other, regardless of what you are, right? Similar cell signaling pathways in diverse eukaryotes are evidence of conserved evolutionary processes. This is true, right? Because no matter what organism you are, again, eukaryotic cell structure is going to be nearly identical, right? How those cells communicate with each other is also nearly identical, right? Cell signaling functions mainly during early developmental stages. That is not true, right? Cell signaling needs to be a process that occurs through all developmental stages in order to sustain life, right? So number 2C. Okay. Number 3. A colony of termites was exposed to an atmosphere of 100% oxygen for 3 days. The insects were not immediately harmed by the treatment, but the protozoa that lived in the termites' guts were eliminated. The treated termites um, continued to behave normally and to eat wood, but they began to starve after a short time. When the treated termites were instead fed wood contaminated with the feces of untreated termites, the treated termites regained the ability to digest wood and no longer starved. The best analysis of the results of the experiment is that. Okay, so just from this situation, we know, right, that the protozoa play an important role in what? Digestion of the wood, right? Um, because again, right, without it, right, they although they eat the wood, they starve, meaning they're not able to break it down, therefore they're starving, right? So we know infection with protozoa causes termites to switch to fermentation, doesn't make sense. Cooperative interactions between the termites and the protozoa allow termites to extract energy from wood and survive. This would be your correct answer, right? A cooperative interaction, more specifically, this is what? Mutualism, right? Protozoa, right? I'm assuming they are, right, breaking down the wood, probably, right, are able to survive due to uh, living in the termites' digestive system, right? And then because of that, the termites are able to extract nutrients for the wood for their own survival, right? Termite immune system eliminates protozoa from the gut. That's not true. Termites digest protozoa as their main source of energy. Also not true, right? Okay, number 4. Ethylene is an organic compound produced by ripening fruits. In a controlled experiment, researchers found that ethylene gas stimulated the ripening process in newly harvested fruits. Which of the following describes the most likely connection between natural ethylene production and fruit ripening? Okay. So ethylene, one of your plant hormones that will come up, right? Pretty often, right? So what do we know about that? Basically, we know, right, that ethylene gas is going to cause ripening in fruit, right? So let's go through the choices. As a result of metabolic inactivity, newly harvested fruits are unable to absorb ethylene from the atmosphere. This is not true because, like they tell you, right? It stimulates ripening, right? Ethylene gas is a chemical signal through which ripening fruits trigger the ripening process in other fruits. This is going to be true, right? Ethylene, if you ripening fruits produce ethylene. Unripe fruits, right? Newly harvested fruits, when they are receive ethylene, I guess, or are exposed to ethylene, they're going to ripen, right? Bananas, right? Bananas are one of the fruit, right, that will release ethylene, right, as it ripens. Okay, because of normal phenotypic variation, only some of the fruits in a given generation are expected to produce ethylene gas. Again, doesn't make any sense. Rate of ethylene gas production by ripening fruits is an indicator of the relative age of an ecosystem. Hopefully no one put D. Okay. Water in a pond contaminated with the weed killer, atrazine, is suspected of inhibiting metamorphosis in northern leopard frogs. A team of scientists collected fertilized northern leopard frog eggs from a different pond that is not contaminated. Which of the following is the best experimental design to determine whether atrazine is responsible for inhibiting metamorphosis in northern leopard frogs? Okay, so how do we set this up, right? Um, so we have a contaminated pond, and then we have frog eggs, right? From a non-contaminated pond, right? So what do we want to do to see if atrazine is going to inhibit metamorphosis? First one, we have placed half of the fertilized eggs in a pool of water with the same concentration of atrazine as a contaminated pond, and place the other half in a pool of water that has no atrazine. Monitor the development of the embryos through metamorphosis into adulthood. This works, right? Half of them have atrazine, and then half of them have no atrazine, right? This is your control, right? Your control group, right? Place all of the fertilized eggs in a pool of pond water with the same concentration of atrazine as a contaminated pond. Compare the number of frogs that reach metamorphosis to those that reach adulthood in the contaminated pond. You have no control. This doesn't work. Allow all fertilized eggs to develop into adults. Expose 1/3 of the frogs to 1/2, whatever. This doesn't work because you're looking at metamorphosis, right? So if they're all adults, doesn't make sense, right? Divide the fertilized eggs into 3 groups and expose each group to a different concentration of atrazine. Release the eggs back into the contaminated pond and check for metamorphosis after 3 months. Okay, again, this doesn't work. You have no control, right? You have different levels of atrazine. This could work if you, or minus the second part, right? But you would need a control group, which would be no atrazine, right? So if I have like different concentrations, like 5%, 10%, 20% atrazine, I would also need a control group that has no atrazine. Okie dokie. Okay, 6 to 10. The food web above represents feeding relationships in a biological community near a deep sea hydrothermal vent. Hydrothermal vents are geysers on the seafloor that gush superheated, mineral-rich water. Seawater surrounding hydrothermal vents typically contains carbon dioxide, molecular hydrogen, hydrogen sulfide, and methane. Sunlight, however, fails to reach the seafloor where deep-sea hydrothermal vents are located. As part of an investigation, researchers collected living specimens from an area near a deep-sea hydrothermal vent. Mussels in the collection were found to be dependent on molecular hydrogen in seawater. Also, the researchers discovered multiple species of bacteria living in the gills of the mussels. Mussels use gills for filter feeding and gas exchange with the surrounding seawater. On the basis of their experimental results, the researchers hypothesized that some bacteria living in the gills of the mussels are capable of chemosynthesis, right? So again, what is chemosynthesis, right? Chemosynthesis is essentially the same thing as photosynthesis, except you're using chemical compounds to make sugar, right? So because you need producers, right? Producers are going to be the base of any food chain, food web, whatever, right? So in order to support life, you need producers. If you have no light, right, you cannot have photosynthesis, right? So chemosynthesis is going to be what happens. Okay, number 6. Which of the following best explains how biological communities near deep sea hydrothermal vents can exist in a habitat lacking sunlight, right? Again, basically, if you have no sunlight, right, you're depending on chemosynthesis, right? Environmental conditions on some distant planets. Okay, that's already weird, right? Heterotrophs metabolize carbon-containing compounds produced by the photosynthetic organisms that live in the seafloor. Again, we cannot have photosynthetic organisms, right? Deep sea hydrothermal vents without sunlight doesn't make sense, right? Some organisms rely on energy captured from inorganic compounds to drive basic biological process, right? This is the definition of chemosynthesis, right? Taking whatever chemicals, right, inorganic compounds, turning it into sugar. Some organisms that can tolerate high temperatures are single-celled, whereas others are multicellular. Again, that is true, but irrelevant to anything, right? Okay, 7. Based on an analysis of the food web, an observation that deep-sea bacteria consume molecular hydrogen is most relevant to resolving which of the following apparent contradictions. Okay. So I guess, right, water gushing from deep sea hydrothermal vents can be hot as 400 degrees, which is lethal temperature for most organisms. That is cool, but nothing to do, right? What is the most likely reason, right, is what they're asking. Why these deep sea bacteria consume molecular hydrogen, right? Some deep sea organisms appear to be primary consumers, but no plants live near the hydrothermal vents, right? This is probably the most likely, right? Your deep sea bacteria, you need to have producers, right? But you have no plants, right? So those deep sea bacteria that are consuming molecular hydrogen are most likely the ones that are your producers, right? They are the ones that are doing chemosynthesis, right? Zoarcid fish are thought to be aggressive predators, but they're frequently described as being lazy swimmers. Oh, not relevant to anything. Some tissues of tube worms contain hemoglobin, but there's little free oxygen at this depth. Again, not sure why that would be relevant to deep sea bacteria. Right. Okay. Researchers are investigating the evolutionary relationship among organisms found near deep sea hydrothermal vents and similar organisms found closer to the ocean surface. Which of the following scientific questions is most relevant to the investigation? Okay. What are the nucleotide sequences of ribosomal RNA genes that are found in the genomes of the different species, right? So again, look ways to look at ancestry. Can you compare nucleotide sequences? Yeah, that works. What large-scale geological events have occurred recently in the mid-Atlantic Ocean? This is not going to tell you anything about evolutionary relationships because, again, they want to know recent, right? Does water temperature at different ocean depths affect the relative levels of dissolved oxygen? Again, how does this have anything to do with evolutionary relationships? Species found near deep sea hydrothermal vents all have the same haploid number of chromosomes. Okay, yeah, that doesn't really say much, right? Having the same number of chromosomes, right, um, is not really going to say much. I don't know why that would do anything. A is the only one that makes any sense, really. Um, and then number 9, on the basis of the food web, which of the following members of a deep sea biological community is most likely to also have a symbiotic relationship with synthetic organisms? Um, so if you look, right, it's going to be one of your the ones at the bottom, right, of your food web, right? I would, let me see what they have. It would probably be, right, it could only really be one of these down here. Mussels, giant tube worms, or shrimp, right? Mussels, giant tube worms, shrimp. Mussels, giant tube worms, shrimp. Mussels, giant tube worms, shrimp, right? This is, shrimp is the only one that's there, right? Because again, your chemosynthetic bacteria are going to be your producers, right? So you want to look at the bottom of your food web, right? Which is shrimp, giant tube worms, mussels. Okay, number 10. To refine their model of deep sea biological communities, the researchers investigated areas of the seafloor that are distant from any active hydrothermal vents. Which of the following is the best interpretation of the observation that some octopus species are only found near active hydrothermal vents? Okay, so you have octopus species, right, that are near hydrothermal vents, right? So why would that be? Is the question. Um, it needs something that from, right, the easiest explanation is that there's something being provided by the hydrothermal vents, or there's another species or whatever that is, right, by the hydrothermal vent that it requires, right? So the first choice, octopuses depend indirectly on inorganic compounds in the seawater surrounding hydrothermal vents. This is likely, right? Organisms capable of moving across the seafloor more difficult to observe than are organisms that are rooted in place. Not sure what that has to do with anything. Genetic bottleneck events in isolated areas of the deep ocean adversely affect some octopus populations, but not others. Again, has nothing to do with that. Biomass in a typical marine food web is expected to be greater in higher trophic levels than in lower trophic levels. This is untrue and irrelevant, so only one that works is A. Right? Biomass is always greater at your lower trophic levels. Biomass gets smaller and smaller as you go up. Okay. On a large volcanic island, researchers are studying a population of annual herbaceous plants. Which of the following observations best supports the prediction that speciation will occur within the existing plant population? Right, so speciation is our macroevolution, right? So individuals of the species sometimes reproduce asexually by producing runners. This has really nothing to do with speciation, right? Lava has separated the population into two areas, upland forest and a lowland marsh, right? So if you separate your species, right, geographically, yes, right? Um, Speciation is very much likely to occur. Multiple groups of birds depend on the fruit produced by the plants as a source of food, has nothing to do with it. Plants produce more seeds during warm summers than they do during cool summers, again, irrelevant. 12. individuals with an inherited autosomal recessive disorder called primary ciliary dyskinesia often have severe respiratory problems due to defective cilia. Males with PCD are often sterile because they produce sperm with defective flagella. Which of the following most likely explains the effect of the recessive allele? Right. So if it affects, right, your flagella, right, um, what is going to be related to defective flagella essentially, right? Mitochondria are defective, not likely, right? It's because it's not that your flagella are can't move, right? Or sorry, um, the reason why this is wrong is your mitochondria have nothing to do with producing protein, right? Um, so incorrect. Plasma membrane of the alveoli is not permeable to carbon dioxide during respiration because it is too hydrophobic. Again, that would be a problem with your plasma membrane, right? So the alveoli in your lungs, again, not sure what doesn't make sense, but also how would that affect your flagella, right? Doesn't connect. Golgi bodies secrete an enzyme that destroys the proteins in the flagella and cilia. Again, Golgi bodies secrete an enzyme, yes, but no, irrelevant. Cells do not produce functional motor proteins in flagella and cilia. This is probably the only one that works. Yeah. Again, you have cilia in your respiratory tract, digestive tract, flagella in your sperm. This is the only one that would connect all of those problems. 13. The diagram below shows energy changes in a specific chemical reaction with and without the addition of an enzyme to the reaction, right? So we know this is going to be an exothermic or exergonic. Exorgonic, I believe. Right? Exergonic. I think it's exergonic. They both look weird. Exergonic process, right? You have uncatalyzed here, and you have your catalyzed reaction, right? The one with a lower activation energy, right, which is the difference from your reactants to your peak, right, is your activation energy. Which of the following questions can best be answered by the diagram? Right, so this is just showing, does your adding an enzyme to your reaction speed it up or catalyze, right? So again, does the activation energy of an enzyme reduce the activation energy required for a reaction? Does the addition of an enzyme result in the formation of covalent bonds? Right? Does the addition of an enzyme produce a greater amount of products? Does the addition of an enzyme change the pathway for the reaction? Right? So all that adding an enzyme does really is it lowers your activation energy. Right? Does it change anything else? No. Right? You still have the same reactants. You still have the same products. It'll just occur quicker because now your activation energy is lower. Okay, 14. The salinity of a small inland lake has recently started to increase. Researchers are planning to study the lake over several decades to investigate how freshwater organisms survive significant changes in their natural habitat. Which of the following physiological mechanisms will the researchers most likely observe among the surviving organisms in the lab? Okay, so again, if your salinity starts to increase, right, then you're going to have adaptations that allow your freshwater organisms survive, right? So they would have to be able to maintain internal salinity separate from external salinity, essentially. Prokaryotic organisms will use various mechanisms to counteract swelling of cells as a result of increased water uptake. If the salinity, right, of your lake increases, right, what is going to happen? Your cells are not going to swell, they're going to shrivel, right? Because if you have a cell, right, in a super high saline concentration, right, then what, according to osmosis, right, what is going to happen? Water is going to move out of your cell, right? So your cell will shrivel, doesn't work. Single-celled Single-celled organisms will use various mechanisms to counteract the increased flow of water from cells to the environment. That is true, right? Again, water will move outwards, not inwards, right? Eukaryotic organisms will use various mechanisms to counteract the diffusion of positively charged ions across the cell membrane. Doesn't make any sense. Multicellular organisms will use various mechanisms to counteract the loss of cell adhesion as a result of calcium deficiencies. Again, none of these make sense. Okay. 15. Assume that there are 50,000 joules of energy available in trophic level 2. According to the conventional model of energy flow in ecosystems, which of the following statements correctly describes the flow of energy in the system? Okay. So we know, right, the 10% rule, right? So from here, right, only 10% goes up, so that's 5,000 joules, right? Up here, it would be 500 joules, which means down here would be 500,000 joules, right? So whichever one of these works, one, this is wrong, one, this is wrong, wrong, the only one, D is correct, right? Always remember 10%. That's why biomass is always greater at the bottom, because only 10% of that energy moves up to the top. Okay, 16. The three-spined stickleback is a small fish found in both marine and freshwater environments. Marine stickleback populations consist mainly of individuals with armor-like plates covering most of their body surface, completely plated. Approximately 10,000 years ago, some marine sticklebacks colonized freshwater environments. After many generations in the freshwater environment, the freshwater stickleback populations lacked the armor plating, low-plated, typical of marine stickleback populations. Over the period between 1957 and 2005, one freshwater population in Lake Washington, a lake in a coastal region of the northwestern United States, changed from having a majority of individuals of the low-plated phenotype to having more individuals of the completely plated phenotype than of the low-plated phenotype. Figure 1 shows the distribution of plated phenotypes in Lake Washington sticklebacks at 4 time points between 1957 and 2005. A single gene, ectodysplasin (EDA), is thought to be responsible for the variation in the number of armor plates in sticklebacks. Figure 2 shows a phylogenetic tree constructed by comparing DNA sequences of the EDA gene from a number of stickleback populations with low-plated or completely plated phenotypes. Figure 3 shows a phylogenetic tree constructed by comparing the sequences of 25 genes that were randomly selected from the same populations as shown in Figure 2. In both figures, shaded populations display the completely plated phenotype. Okay. So you can see here, right? Complete, partial, low plated, right? So in Lake Washington, what do you see? You see partial plated. Majority are gonna be low plated, right? And then you can kind of see it even out where you have in 2005 you have more completely plated, partially plated, and then not very many low plated, right? So you kind of see a shift, right? And then the gene, right? You have two phylogenetic trees. First one is based on the EDA gene, and then this one is based on random genes, right? So you can see here, right, completely plated are going to be, when you just look at the EDA gene, right, you see you're completely plated on one clade, low plated on the other. Okay, 16. Which of the best explains the differences in the armor of the Lake Washington stickleback population summarized in Figure 1? Uh, oh, here, right? Okay, so kind of let's just look through the answers. Analysis of somatic cells using chromosomal staining and light microscopy indicates that stickleback fish have a diploid number of 42. Again, completely irrelevant. Stickleback males from natural freshwater populations are typically more aggressive when competing for mates than are stickleback males from laboratory bred populations. Again, I'm not exactly sure what this comparison has to do with the figure one, right? Fish exhibiting the low-plated phenotype were selected against in the Lake Washington stickleback population over the last 50 years. We know this is true, right? They were selected against. That's why you saw a shift, right? From the, if you look at figure one, right? Like about 90%, right, were low plated. And then in 2005, you had less than 20%, right? So what does that mean? Being low plated became disadvantageous, right? So you can see a shift in a, or you can see a huge decrease in low plated, and your partially plated and completely plated increase, right? And you can see that over time, right, which one had the best advantage? The ones that were completely plated. Because if you look at 2005, right, you have the highest percentage of fish completely plated, right? So the answer will be C. Again, migration um, could have happened, right, to the lake, but again, not sure what that has to do with anything, right? Okay, 17. A completely plated stickleback from a marine population was mated to a low-plated stickleback from a freshwater population. The resulting F1 hybrids all displayed a completely plated phenotype, right? Okay. When the F1 hybrids were allowed to interbreed, the resulting F2 generation included completely plated offspring and low plated offspring in a 3-to-1 ratio. Which of the following conclusions is best supported by the results of the breeding experiment? Okay. Well, first of all, if you take completely plated and low plated and you breed them and you get all completely plated, what does that tell us? Completely plated is dominant, low plated is recessive, right? That explains again the 3-to-1 ratio, right? If I have big P crossed by little p, right? Then my F1 generation would all be big P, little p. And then my F2 generation, right? I would have a 3-to-1 ratio, right? So let's see. That would be B. Oh, controlled by many. Armor loss is an acquired characteristic that is affected by one or more environmental factors. Again, it doesn't make sense. Acquired characteristics also cannot be passed down, so it doesn't make sense anyways. Um, Patterns of armor plating in stickleback populations are regulated by sex-specific signals. No evidence of that whatsoever. Okay. Um, Number 18. Prior to 1960, Lake Washington was highly polluted and underwater visibility was limited to 1 or 2 meters. In the late 1960s, a large cleanup effort reduced pollution, resulting in visibility increased to 6 to 7 meters by 1976. Which of the following best explains how the change in underwater visibility affected armor plating in Lake Washington sticklebacks between 1957 and 1976? Okay, so 1957, it was dirty. 1976, water became clear, right? Probably had to do with visibility of the predator, right? So you see low plated when your lake was dirty. As your lake got clearer, right, and cleaner, right? You see fewer low plated and then more plated, completely plated, right? So we know that the cleanliness of the lake, right, had to do with what's it called? The plating of the sticklebacks, right? So as the water got clear, it's probable to assume, right, that they were easier to spot by the predators, right, um, and therefore having more plating became beneficial, right? So let's see, higher visibility allowed sticklebacks to increase their food consumption to make armor production easier. No. Higher visibility made the sticklebacks more susceptible to large-toothed predators in the lake, giving complete armor a selective advantage. Yes. Right. Clear water allowed sunlight to penetrate to deeper depths, so the sticklebacks had to increase the amount of armor to protect themselves from the resulting increase in water temperature. No. Clear water contained fewer of the molecular building blocks needed for armor protection, so stickleback showed a reduction in the amount of armor. This is so wrong in multiple ways. Okay, 19. Evolution of a new trait typically takes many generations. Yet, a dramatic shift in the extent of armor plating in the Lake Washington stickleback population occurred in the 50 years following the cleanup of the lake. Which of the following best describes the mechanism of rapid evolution of the armor phenotype in the Lake Washington sticklebacks? Okay, so it's really an example of microevolution, right? And what is microevolution? A change in the allelic frequencies of the population, right? So the frequency of the dominant allele in this case, which is armored, right, or armored plating, right, went from very rare, right, to very more common, right. So anytime again the frequency of your allele changes, we call it microevolution, right. So what happened, right? Pollutants forced the sticklebacks to increase their mutation rate. Does not make sense. Rapid change in the Lake Washington ecosystem required individual sticklebacks to evolve complete armor quickly. That's individuals do not evolve. What is the smallest unit on which evolution can occur? Those are your populations, right? Only populations can evolve, right? That is the lowest unit where evolution is possible. The increase in visibility in Lake Washington allowed sticklebacks to visually select mates with low armor, right? Again, right? Probably not, right? Um, a change occurred by mate choice, not very likely. Selective pressures favored individuals with the plated phenotype, causing the plated allele frequency in the population to quickly increase, right? Yes, right? The new selective pressure, which is what? The water clearing up, increasing visibility for the predators, making your sticklebacks more prone to predation, right? So having that armor helps, right? So D. The phylogenetic trees in figures 2 and 3 depict two different phylogenies of the same population of sticklebacks. Which of the following questions will best help determine which tree represents the most accurate phylogeny? Okay. Um, we have two, right? Again, which of the following questions? Yeah. Okay. So we have one based on EDA gene. We have one based on 25 random genes, right? And you can see, right, that you get two completely different phylogenetic trees, right? So what is a valid question we can get? Is the EDA gene as representative of the differences between the populations as the 25 random genes that were examined for figure 3? Yes, right? Um, I mean, yeah, if you just compare the EDA gene, right, you get something like this, right? But is the EDA gene something that can represent different major differences in your fish, right? Can it be representative of your differences, right? Can one gene be representative of all of your differences, right? Um, I don't know. B, I think B doesn't really make sense, right? Are the low plated populations found only in freshwater? High plated populations found only in saltwater environments? Again, not really sure how this has to do with phylogeny. Is the common ancestor of the organisms represented in Figure 2 different from the common ancestors of the organisms represented in Figure 3? Um, I mean, if you look at it, right, obviously they're not, right? That's the easily answered question, right? Doesn't tell us, right? Um, doesn't help us decide which one is better, right? Is the expression level of the EDA gene expressed analyzed in Figure 2 significantly greater than the expression levels of the 25 genes analyzed in Figure 3? Expression level of your genes has really nothing to do with anything, right? So best answer will be A. Okay. Number 21. Which of the following occurs in all species of living organisms and may lead to an increase in genetic mutation? So, what can increase genetic variation? Mutations in the genome, yes, right? Does that occur in all species of living organisms? Yes. Crossing over in meiosis. Does meiosis occur in all species? No, right? So that doesn't work. Random assortment of chromosomes? No. Alternative splicing of mRNA? No. 21A is the only one, right? 22. Researcher examining a root tip observes a plant cell with condensed sister chromatids, kinetochores with attached microtubules, and individual chromosomes that are aligned at the equatorial plate of the cell. Which of the following best describes what the next process will be in the cell? Right? So basically we have what? Uh, mitosis, right, occurring, right? So what happens after, right, they line up, right? Your chromatids are going to separate, right? And they're going to move towards, right? Basically, we're at metaphase, right? So you just need to describe anaphase, which is going to be B, right? Paired chromatids separate, new daughter chromosomes will move toward opposite poles of the cell. How do we know it's mitosis, not meiosis? We're looking at a root tip, right? Root tip, we will have mitosis because what is your root doing? Uh, it's going to be growing, right? Extending into the dirt, right? Okay, 23. The figure below illustrates a eukaryotic cell. Which of the following best describes how the three structures indicated by the arrows work together? So we have Ruffyar, Golgi apparatus, vesicle, right? What does the Ruffyar do? Produces proteins, right? Usually going to produce proteins that are meant to be shipped outside or for your cell membrane, right? Free ribosomes usually are going to produce proteins that remain within your cell, right? So Ruffyr usually makes your export or membrane proteins, right? What happens after your Ruffyr makes those proteins? Moves to the Golgi apparatus. Golgi apparatus is going to do what? Modify and package those proteins, and they will package it into a vesicle, right? That buds off of your Golgi apparatus and can be exported to your cell, right? So the answer that works: B, synthesize and isolate proteins for secretion or for use in the cell, right? Again, A, C, D do not work, right? Um, this A, right? This is done by your smoothie R. Catabolize nutrients and produce ATP for intracellular energy storage, right? ATP is mitochondria, synthesize all ribosomal proteins, right? Yes, you're going to do that, but you don't need your Golgi vesicle, Okay, 24. In the following human pedigree, squares represent males, circles represent females, and shaded symbols indicate individuals affected with the disorder. One of the affected males from the third generation has a child with a female who is a carrier. For the pedigree shown above, which of the following best expresses the probability that the couple's first son will be affected with the disorder? One of the affected males from the third generation. Okay. So it's either this one or this one. Doesn't really matter, right? Um, so what? First of all, is this a dominant disorder or recessive disorder? We know it's going to be a recessive disorder because two healthy individuals, right, can produce someone who is not, right? Um, is this going to be sex linked? If you look, right, looks pretty sex linked to me, right? Because you see that only males are affected, right? Um, now, if this female is a carrier, makes sense, right? This female is a carrier makes sense because in order to have two males, right, mom has to be a carrier, right? So if you have a diseased male, right, crossed by, oh, sorry, that is not a male, diseased male crossed with carrier mom, right? What's going to happen to their sons, right? Right, 50% will have it. Right, so 24 it will be B. Chytridiomycosis is a potentially lethal fungal infection that adversely affects some frog populations. The incidence and severity of the fungal infection can vary over time. Which of the following statements best describes the changes in the frog population that are depicted in the model above? Okay, so you can see you have two y-axes, proportion of population infected and relative population size, right? So you can see your population size goes down, up, right? And proportion of population affected is essentially inverse to your graph. So why do we see that? Or what explains that, I guess? The fungus that infected the frog population was initially virulent, but it later became harmless. Based on the trend over the past few years of the study, the fungus has become extinct, right? There's no evidence for this. And we do know that what happens is as you have more infected, right, your population size decreases. So it is lethal for sure. Infected individuals gradually died out and genetically resistant individuals became more common. The frog population recovered because of increased frequency of resistant individuals. Uh, hold on, sorry, give me 1 second. Okay. Infected individuals gradually died out, and genetically resistant individuals became more common. The frog population recovered because the increased uh, frequency of resistant individuals. That would explain, right, why here, right, um, your population size decreased and you had more infections, but like here you kind of had a change, right? Where your proportion of your population decreased and then your population size recovered, right? That means that you had likely had more resistant guys. Patterns indicate a sudden increase in the severity of the fungal infection. Large proportion of the frog population died because they could not evolve fast enough. Not true. Size of the frog population decreased sharply upon initial contact with the fungus. Fungus eventually became symbiotic, and the frog population began to recover. Likely not, right? So knocked a bunch out, and then it was able to recover because again, um, yeah, resistance. Okay. 26. The trip operon is a coordinately regulated group of genes, TRPA to TRPE, that are required for tryptophan biosynthesis in E. coli. Based on the figure above, which of the following correctly describes the regulation of the trip operon? Okay, so remember, what do we know about the trip operon, right? It's on until turned off, right? We call it a repressible operon, right? Lac operon is inducible, right? Off until turned on, right? Um, so if you didn't remember that, just look here, right? You're an active repressor, right? What happens in the presence of tryptophan? Tryptophan is going to activate your repressor and it's going to block transcription, right? Again, your operon is on until you turn it off. How do you turn it off? If you have a lot of tryptophan present, right? Do you want to keep making tryptophan? No, you're going to turn it off, right? So it's going to be B in the presence of tryptophan, repressor is active and binds to the trip operator, preventing RNA polymerase from transcribing the option, right? So we call tryptophan a co-repressor, right? Because again, it's going to activate your repressor, block transcription. Okay, 27. Uh, the pesticide DDT was widely used in the 1940s as a method of insect control. In the late 1950s, the first DDT-resistant mosquitoes were discovered, and eventually DDT-resistant mosquitoes were found globally. When DDT is used now, the development of DDT resistance in mosquito populations occurs in months rather than years. Which of the following best explains the observations concerning DDT resistance in mosquitoes? Okay. So basically, right? DDT resistance occurs quickly, right? What does that mean, right? This is again an example of natural selection, right? Having DDT resistance is a selective advantage, right? So let's go through. Competition for limited resources already sounds wrong. Causes mosquitoes to migrate to geographical areas that have richer supplies of DDT. Does not make sense. Proportion of DDT resistant mosquitoes in a population remains constant due to the metabolic cost of DDT utilization. Again, doesn't make sense. Natural selection favors DDT-resistant mosquitoes that are already present in a population when DDT exposure occurs. Yes, right? Again, if you have DDT resistant mosquito already, right, those are the ones that are going to survive. And because they're the ones that are going to survive, they're going to leave more offspring, etc. DDT is a chemical signal that delays normal reproductive cycles in many mosquito populations. Does not make sense. Okay. 28. Two nutrient solutions. Two nutrient solutions are maintained at the same pH. Actively respiring mitochondria are isolated and placed into each of the two solutions. Oxygen gas is bubbled into one solution. The other solution is depleted of available oxygen. Which of the following best explains why ATP production is greater in the tube with oxygen than in the tube without oxygen? Okay, so basically, we know that ATP production is being done, right? Um, yeah. Um, I maintain at the same pH, blah, blah, blah. Okay. So they're already doing, right, uh, respiration, right? So let's look through the answers. Rate of proton pumping across the inner mitochondrial membrane is lower in the sample without oxygen, right? Remember, if you have no oxygen, what happens to your ETC? It stops, right? Why does it stop? Oxygen is essential in your ETCY. It is your final electron acceptor, right? If you have no final electron acceptor, right, your electrons are not being pulled through, right? Therefore, you're going to be making essentially no ATP, right? You're going to have to resort to fermentation, right? So A. Um, plasma membrane is not the problem, right? Oxidative phosphorylation produces more ATP than does fermentation. Not true, right? Oxidative phosphorylation only occurs with oxygen, right? Fermentation is going to be what's happening to the mitochondria that have no oxygen. Okay, so glycolysis does not require oxygen, remember, right? So both of those two mitochondria are going to be making the same amount of ATP just from glycolysis, and you don't need oxygen for that. Okay, table 1 shows the results of breeding experiments to examine the inheritance of flower color, purple versus white, and pod shape, inflated versus constricted. For the crosses recorded in table 1, true breeding parents were crossed to produce F1 offspring, which were then test crossed to homozygous recessive individuals. Table 2 shows the results of computer simulated crosses to model the inheritance of leaf shape, broad versus narrow, and flower color, purple versus white. Okay, so table 1, right? So they kind of show you, right, your test cross, right? True breeding, so that means purple, they're both homozygous, right? And then table 2. Based on the data in table 1, which of the following best explains why there are no individuals with constricted pods in the F1 generation? Right, so table 1, F1 generation, right? Why do you only have inflated? Well, that tells us that inflated is dominant to constricted, right? So when you take homozygous dominant type crossed by a homozygous recessive, right, you're only going to get heterozygous individuals, right? And if the phenotype is inflated, that means that inflated is completely dominant to constricted, right? So something along those lines would work. Inflated pod shape is dominant to constricted pod shape, so A. Okay, 30. In table 1, the ratio of phenotypes in the offspring from the test cross of F1 plants that had purple flowers and inflated pods suggests that the genes for flower color and pod shape are located. Okay. Uh, purple flowers and inflated pods. Purple flowers and inflated pods. Purple flowers and inflated pods. So this, right? So the phenotype was purple and inflated, right? And if you look here, right, you had purple inflated, white constricted, right? Purple inflated, white constricted, right? If you have more that are purple inflated and white constricted, then your new recombinant, right? Recombinants would be purple constricted and white inflated. It would indicate that you have what? Gene linkage, right? Um, Here, right? What do we see, right? You don't see linkage, right? Because what would happen if the genes were located on the same chromosome or very close to each other, right? The majority, right, would have the phenotype, right, of your original, of your parents, right? So we don't see that, right? So we know that they're going to be on different chromosomes. C. Okay, which of the following provides the best justification for an assumption that might have been used in the computer simulation? Table 2. Okay, so table 2, right? Broad, narrow, you got broad, purple and white, you got purple, right? Broad, white, narrow, purple, you got broad and purple, right? Again, dominant, dominant, right? When you cross those, you got broad, white, purple, broad, purple, narrow, purple, right? So what does this test cross tell you? You can see linkage here, right? 664, majority are going to be have the same as your parental cross, right? Um, let's see. Broad allele is recessive. Okay, no, this we know broad is dominant. Purple allele is dominant to the white allele. Yes, that is true because all of the offspring from the cross of purple flowered and white flowered plants have purple flowers. Okay, that is true. Narrow allele is co-dominant with the purple allele. That doesn't make sense, right? You're looking at two different traits. They cannot be co-dominant. White allele is dominant to both the broad and narrow alleles. Wait, that doesn't make sense. B is the only one that makes sense. 32. In table 2, the F1 offspring of the cross between broad-leaved white-flowered plants with narrow-leaved purple-flowered plants have a phenotype that differs from that of either parent. However, many test cross offspring have the same phenotype as one of the two plants in the parental cross, but relatively few test cross offspring have the same phenotype as the F1 offspring. Which of the following best explains the observation? Okay. Okay. Okay, so it's looking at this, right? So how come when you took broad purple, cross it with broad purple, right? You barely got any broad purple, right? You got broad white, narrow purple, right? That's just what happens, right? As a result of recombination, right? Um, which of the following best explains the observation? Let's see. Recombination between the leaf shape and flower color genes resulted in chromosomes carrying a dominant allele of both genes. Um, yeah, that did happen, right? Because again, you have independent assortment or independent segregation of your leaf shape and your flower color, right? So yes, recombination between the broad and narrow alleles of the leaf shape gene resulted in chromosomes carrying three different alleles. Doesn't make sense. Independent assortment of homologous chromosomes resulted in the combinations of alleles present in the parental generation. Um, yes, that is true. However, that doesn't show you why you have different uh, phenotype, right? Computer model cannot capture, again, doesn't make sense. Okay, 33. When a stimulus is applied to a receptor in the skin, an action potential is propagated along a neuron to the brain, where another signal is sent back to the muscle for a response. Which of the following best describes what occurs when the action potential reaches a chemical synapse at the end of an axon? What happens when, remember, if I were to draw this, right? So action potential, right? Saltatory conduction. What happens at the end, right? It opens calcium ions. Calcium ion flows into your axon terminal, right? And then what happens? Your neurotransmitters located in vesicles move to your synapse. They open, releasing neurotransmitters into your synapse, right? And then those neurotransmitters are picked up by whatever your axon is connected to, right? And then any leftover neurotransmitters is going to be taken back in, right? You're going to do reuptake, repackage it in the vesicles for your next action potential, right? So D. Yeah. Okay. 34. Methicillin-resistant staph MRSA can be a serious threat to human health. There is evidence that S. aureus infections are common in hospitals and that MRSA have become resistant to other antibiotics besides methicillin. This suggests that the rapid evolution of resistance in the bacteria poses a serious public health challenge. Which of the following best explains the ability of MRSA to evade existing drug therapies? Okay. So essentially, right, you have they're saying that this bacteria, right, um, is no longer just uh, resistant to methicillin. It's resistant to other bacteria, right? This is an example of what? Horizontal gene transfer, right? Remember why a lot of like super bacteria or super bugs, a lot of them develop in hospitals. And the reason why they develop in hospitals is because in a hospital you have so many different bacteria that are resistant to different antibiotics, right? Now, if they're together, right, what can happen is your horizontal gene transfer. What are your horizontal gene transfer? You have transformation, transduction, conjugation, right? So what does that mean? Gene resistance, right, or a gene from one bacteria can be transferred to another bacteria through horizontal gene transfer, right? And therefore, right, um, you can have bacteria that are resistant, right, to multiple antibiotics without it having to develop that resistance on its own, right? It's getting the gene for the resistance without having to have, I don't know, gotten it through naturally, right? Um, so let's see. Whoa, what happened? MRSA have long generation times and very large population sizes. Okay, cool, irrelevant. MRSA develops new alleles by intentionally. You cannot intentionally introduce mutations, right? All mutations are random, right? Um, you can use like mutagens to induce a mutation, right? But you cannot pick what mutation you're inducing. MRSA metabolize many drugs in their lysosomes and therefore evolve resistance at a high rate. Irrelevant. MRSA exchange genetic material with other antibiotic-resistant bacteria, which can spread resistance in the S. aureus population. Yes, right? Horizontal gene exchange, right? Whether that's transformation, transduction, conjugation. Okay, 35. Researchers conducted a controlled experiment to investigate the effect of diet on the health of beef cattle. Initial hypothesis was that an abrupt change in diet will benefit beef cattle by reducing the sizes of bacterial populations living in the digestive systems of the cattle. In the experiment, the researchers determined the relative abundance of two bacterial species found in the rumen of cattle. The rumen is a part of the stomach that acts as a fermentation chamber in cattle and other ruminants. Shown in the table are the results from before and after an abrupt change in the cattle's diet. Based on the results, which of the following best explains why the initial hypothesis should be revised? Okay, so it says that if you abruptly change their diet, right, it reduces the size of bacterial populations, right? So if you look, right, principal diet was hay, they changed it to gray, right? Um, what happened after, right? You see high, right? Streptococcus bovis, but then you see low, right? So it doesn't really work, right? This, right? So you expected it to reduce both, right? But it increased one, right? So obviously you need to adjust, right? Um, so it's going to be B. Abundance of one of the bacterial species increased in response to the change in diet, right? So it did not do what they had expected. The table below describes the action of two genes involved in the regulation of nervous system development in the nematode C. elegans. Which of the following claims is best supported by the data? Okay, so gene A, gene B, when they're both inactive, no neurons. Gene A is inactive, B is active, no neurons, right? A is active, B is inactive, you have way too many neurons, right? Both are active, normal number of neurons, right? So what do you think that gene A does? Probably promotes neurons, right? Growth of neurons. B does what? Slows it down, right? Because, or not, like removes neurons, right? Because if you only have gene A, you have way too many neurons, right? If you just turn on gene B, nothing happens, right? So gene A is promoting neuronal development. Gene B likely promoting programmed cell death, right? That's why you need both of them on in order for a normal number of neurons to develop, right? So that is A. Okay, 37. Which of the following diagrams best represents hormone-activated gene expression, right? What do we know about hormones, right? Hormones are chemical signals, right? Depending on the actual hormone, right, it may or may not be able to enter your cell, right? This one doesn't work. This one doesn't work. They're not hormones, right? A and B are the only ones that are going to occur. We're looking at gene expression. This is doing apoptosis, right? Doesn't work. Right here, right? Steroid hormones, right? Like estrogen and testosterone, right? They're what? Made of cholesterol, right? So they can enter, right? Because they're nonpolar, right? They can enter your cell membrane, right? So you don't necessarily need a receptor for for your steroid hormones because they can go through your cell membrane, right? So it's going to go through, right, attached to your receptor, which is an intracellular receptor, right, rather than an extracellular receptor that you see for peptide hormones, which is why this is wrong, right? Peptide hormones require an extracellular receptor like a GPCR, right? Um, and then that hormone receptor complex is going to enter your nucleus, right? Initiate transcription, lead to translation. Again, your steroid hormones can go through any cell membrane, right? Including your nucleus. So 37 is A. Okay. The bacterium Vibrio cholerae is harmless unless a lysogenic bacteriophage provides the gene coding for the cholera toxin, which converts the bacteria to the virulent form that causes cholera. Which of the following best explains how the gene encoding cholera toxin becomes part of the bacterial genome, right? So this is just basically a question of how does bacteriophages, which is what? A virus, um, affect a cell, right? Cholera or bacteria, right? So how does your virus affect bacteria, right? It inserts its gene into the host, right? And then the gene is expressed, with the rest of the host cell's genes, right? Lysogenic cycle, right, of your bacteriophage. And bacteriophages are viruses, don't forget. A species of snail lives in the intertidal zone along the coast of New England. The dark-colored variety of the species is more common in northern New England. The light-colored variety is more common 200 miles away in southern New England, and both varieties are commonly found together in central New England. Which of the following best explains the observed distribution pattern of the snails? Okay, so why do you have such a difference? Um, let's see. The founder effect suggests that dark-colored snails migrated from the southern regions to the north and established the populations found there. Huh. I don't know. You see both in central, right? Doesn't really make sense. Genetic drift at the shell color locus caused the northern population to become homozygous with the dark color. Definitely not, right? Mutation rate is higher in the snails as the longer days expose the snails to more ultraviolet radiation than in the north. Doesn't make sense. Dark colored snails absorb more solar energy and so survive more readily in colder northern waters. That would explain why you have more dark colored snails in the north, fewer, and then you have light colored in the south, where it is warmer, right? Central England, you have both. See? Weird question. Okay, which of the following representation best shows a portion of an axon at rest before or after an action potential? Okay, so we have what? Plasma membrane, and then I'm assuming this is a pump. I don't understand. This is outside, this is inside, right? We have what controls it: sodium influx, potassium efflux, right? Sodium influx, potassium efflux, right? So A and B. What is the difference between A and B? Does anyone see a difference between A and B? I'm like tripping out. Oh. No shit. Obviously it's A. Wait, I was like tripping out. I was like, what is the difference? I didn't look carefully enough, right? Sodium influx, potassium efflux, so A. Yeah, sorry. My bad. I was focused on the plus minuses. Okay, hormonal control of male reproductive system. Okay, so you can see, right? Um, right, pluses mean promote, minus means inhibit, right? Your hypothalamus releases GnRH, your anterior pituitary releases LH, luteinizing hormone, FSH, follicle stimulating hormone, right? LH is gonna promote your Leydig cells, which activates your Sertoli cells, which activates your germ cells, and ends in sperm production, right? Now, you don't, in order to stop it, right, your Leydig cells or testosterone, right, is going to inhibit all of these, right? Inhibitin will also inhibit your anterior pituitary. Precise regulation of specific hormone levels is required for optimal sperm production in mammals, as summarized in the figure above. Anabolic androgenic steroids, AAS, are synthetic variants of testosterone that are sometimes abused by persons who desire to enhance their athletic performance or alter their physique. Assuming that AAS would function in the same way as naturally occurring testosterone, it is most likely that long-term abuse of AAS would. Okay, so what does testosterone do? It inhibits your anterior pituitary and your hypothalamus, right? So what would that eventually cause? Decrease in your sperm production, right? Because again, it's going to, right? If you block your hypothalamus and your anterior pituitary, right, or inhibit it, you're basically inhibiting everything downstream from that, right? So you will have a reduction in the amount of sperm that you make. So 41 will be D. Okay. Wolves, a top predator, were reintroduced to Yellowstone National Park in 1995 after a 50-year absence. In a multi-year study, the numbers of wolves and elk were monitored. The data are shown in Figure 1. In two different environments, scientists monitored the percent of aspen trees browsed by herbivores as well as the growth of the trees. The upland environments consist mostly of flat forested areas. The riparian environments are areas along streams with steep wooded banks. Okay, so you see, right? Elk population, wolf populations, they're inverse, right? Which very likely means that elk are the prey of wolves, who are the predators, right? In Yellowstone, right? Browsing of aspen, right? Again, you see that browsing of aspen, again, you see this pattern, right? Reverse pattern in aspen versus growth. So what does that mean? The more you browse your aspen, right, um, the less it grows, right? It's also inverted, right? Um, Based on the data, which of the following is the best explanation for the changes in the elk population size between 2000 and 2005? Okay, so 2000 and 2005, 2000, 2005, right? So why did the elk population decrease or wolf population increase, right? Yeah, predation by wolves was higher than before 1995, right? If you have more predators, right, your prey will likely decrease. Predictions about the community is most likely true. Okay. Decrease in the elk population will cause wolves to feed on aspen trees. No. Increase in the wolf population will lead to a decrease in aspen growth. If you look at the graph, right, aspen growth, right, what do you notice? Right, aspen growth increases when you have a decrease in elk, right? So that means that, let me see, let me look back. Browsing of aspen. Growth of aspen. Yeah. Increase in the growth of aspens will lead to a decrease in the wolf population. If you have increase in the growth of aspens, then what should you have? More elk, which means more wolves, right? Decrease in the wolf population will lead to a decrease in the mean aspen height. This is true, right? Why? Less wolves, more elk. More elk mean they're gonna eat the trees, right? Decrease in mean aspen height, right? Because that's the relationship that you see between your elk and your tree growth, right? The more elk you have, the less your tree, the mean average height or the height of your aspens increases. Okay. Based on the data, which of the following behaviors in elk could account for the differences between the percent of aspens browsed by herbivores and the height of aspen trees in riparian and upland um, environments, right? So essentially, you have riparian and uplands, right? So what your elk, right? What do they like to? They prefer uplands, right? Because when you have a lot of wolves, right? They prefer uplands. right? Or browsing in the uplands, right? Over riparian. What are uplands? Uplands is flat forest. Riparian are long streams with steep wooded banks, right? So they would prefer, right? Um, elk tend to avoid riparian areas with the steep wooded riverbanks, make it difficult to escape predators. Right? Yes. A. Right? If you have a lot of wolves, we don't go there, or the elk don't go there. Okay, 45. Which of the following models best predicts the effect on the ecosystem if a deadly communicable disease is introduced into the wolf population? Okay, so you would expect your wolf population to decrease. What would happen to your elk population? It would increase, right? Which is A, right? Again, no predator, then your elk are gonna have a huge population growth spurt. Okay, information is transmitted through the nervous system when one neuron signals another neuron. The structure of neurons enables transmission to proceed quickly and efficiently. Which of the following diagrams correctly identifies both the structure of neurons and the direction of information flow between neurons? Right? Okay, so it starts at your what? Any information, it starts at your dendrites, moves to your axon terminals, right? This is not correctly. You see your soma, right? It has to be inverted. Looks like this, but this is wrong. So dendrites to axon terminals D. Whoa. Right, remember, these are your dendrites, these little spiny things. This little center, that's your soma or your cell body, right? Axon here, and then your axon terminals here. And then your axon terminals are going to either connect to another dendrites. If you have a neuromuscular junction, it's going to connect to a muscle, etc. But yeah, information through neurons is always one direction, right? It cannot go backwards. Okay, 47. A series of crosses is performed with fruit flies to examine inheritance of the genes vestigial and cinnabar. The recessive VG allele causes small malformed wings called vestigial wings. The recessive CN allele causes bright red eyes called cinnabar eyes. In the first cross, a female with wild-type wings, right, so normal, and eyes is mated with a male with vestigial wings and cinnabar eyes. All the F1 individuals have wild-type wings and eyes, right? Again, you take homozygous dominant, homozygous recessive, F1 will always be heterozygous, right? In the second cross, female F1 flies are mated with males with vestigial wings and cinnabar eyes. Phenotypes of 500 individuals are shown below, right? So what do you see, right? You should see 9331, right? If you do a heterozygous dihybrid cross, right? Is this 9331? No. Everyone is like the either parent, right? So, right? So what does that tell us in this case, right? They're linked. We have gene linkage, right? So that means they're linked on an autosome, right? They did not separate, right? Your wing type and eye color did not successfully separate. Right, and you have a fairly rare, only about like 51 out of 500, so that's like only 10%, right, recombinance. So that means your genes are located pretty close together. Okay, 48. The graph above shows changes in glucagon and insulin secretions at different concentrations of blood glucose. Which of the following feedback mechanisms is best supported by this data? Okay, so glucagon secretion, right? You have that. Um, so what happens? Glucagon and insulin, right? They're opposites, right? When you have high glucose, insulin brings down, right, your blood glucose, And then when you have low blood glucose, what happens? Your glucagon is released to bring up your blood glucose, right? So again, in order to maintain homeostasis, when you have low blood sugar, glucagon makes it go up. When you have high blood sugar, insulin makes it go down, right? So the answer to this is C. Right? Low glucose causes release of glucagon, which stimulates the release of more glucose from tissues, which in turn lowers the amount of glucagon being released, right? High levels of insulin cause greater sugar absorption by your cells, which is then going to lower your glucose levels, right? So they're doing opposite things in order to maintain homeostasis. Okay, 49. Which of the following best describes an advantage that eukaryotic or eukaryote organisms have over prokaryote organisms? They lack a cell membrane, are unable to control what enters or exits the cell. Not true. Um, Eukaryotes have a nuclear envelope separating their DNA from the rest of the cell, which increases the likelihood of advantageous mutations. Not true at all. Eukaryotes have mitochondria and chloroplasts that contain their own genome, which allows the cells to reproduce more rapidly. First part is true, but it has nothing to do with reproduction. Eukaryotes have organelles that allow for compartmentalization of cellular processes, which increases the efficiency of those processes. Yes. Right? Remember, prokaryotes have no membrane-bound organelles. 50. Gregor Mendel's pioneering genetic experiments with pea plants occurred before the discovery of the structure and function of chromosomes. Which of the following observations about inheritance in pea plants could be explained only after the discovery that genes may be linked on a chromosome? Gene linkage, right? See? Most offspring of a given cross have a combination of traits that is identical to either one parent or the other, right? He was lucky that not those traits were not linked. 51. In animals, the Hox genes encode a family of transcription factors that are important for proper development of embryonic segments and are widely conserved in organisms. Figure below shows the embryonic segments in which one such gene, HOXCX, is expressed in the embryo of a mouse, a chicken, and a goose. Embryonic segments are counted from the anterior end. During the formation of vertebrae, the most anterior embryonic segment that expresses HOXC6 marks the end of the cervical neck vertebrae and the beginning of thoracic rib vertebrae. All mammals have 7 cervical vertebrae. Which of the following is most likely to be true? Okay, so whoa. All mammals have 7, right? So 3, 6, 7, 3, 6, 7, right? Most HOXX is expressed, right? So this is your embryonic segment. Marks the end of the neck and the beginning of the thoracic, right? Marks the end of the neck, beginning of your thoracic, here, where your HOX X6, HOX C6, right? So which of the following? Chick and the goose have the same number of thoracic vertebrae. Chicken, goose? No. Chick has what? 6, 7, 3, 6. Goose has 8. Most anterior expression of HOX C6 is the 8th vertebrae in mammals, right? So that would be 1, 2, 3, right here, right? That is the only mammal that you have, right? So let me see. That would have to be, right? Because it's the end of your vertebrae, beginning of your thoracic rib vertebrae, right? So this would have to be your 8th vertebrae in mammals, right? HOXCX is expressed in the same embryonic segments in birds and mammals, right? So you have a bird and a mammal. That's obviously not true. HOXCX is expressed in the same vertebrae at the interior end of all bird embryos. Again, if you look at these, also not true, right? Only one that works is B. The process depicted in the image above is best summarized by which of the following descriptions, right? You have your chiasma, right? Your paired homologous chromosomes, right? So this is what? synapses, right? In what happens during synapses, crossing over, right? During meiosis, crossing over leads to recombination of alleles between homologous pairs of chromosomes, right? When do your homologous chromosomes pair, right? Only during meiosis, right? Prophase 1 of meiosis, where crossing over occurs. Okay, let's go on. A biologist is studying territoriality in 3 related species of birds found in the same section of a forest. For each observed pair of nesting adults, the biologist measured the distance to the next closest nesting pair of the same species, nearest neighbor distance. An analysis of the results is shown in the table below. Which of the following would be most appropriate for identifying the species with the most even distribution of nests? Okay, so 1, 2, 3, mean nearest neighbor, okay, range. Let's see, in order to find it, to find distribution, probably standard deviation, right? What is the average and how much do our values deviate from the average, right? If you have a very low standard deviation, that would tell you that it's what? Fairly uniform, right? If you have a really high standard deviation, that means it's what? Clumped or random, right? So C. Okay. Researchers observe a large population of birds on a remote island. Birds in the population are found to have either red crest feathers or white crest feathers on their heads. Genetic analysis indicates that the allele for red crest feathers is dominant over the allele for white crest feathers. In a survey of the population, the researchers determined the frequency of the crest feather phenotypes. The results of the survey are shown in the table below. Phenotype: red crest feathers 11,000, white crest feathers 1,800. Assuming that the population is in Hardy-Weinberg equilibrium, what proportion of future populations is expected to be heterozygous controlling it, right? Okay, so we know this is dominant, this is recessive, right? So we know that 1759, right, out of 11088 plus 1759 is equal to q squared. Let me calculate this real quick. 11088 plus 1759. 1759 divided by 12847. So q squared equals 0.137. Right? So what is q? Hello. Square root of that. So q equals 0.37. So so p equals 0.63. Right? So they want the proportion of heterozygous, right? So that's 2pq, which equals 2 times 0.63 times 0.37. 0.63 times 0.37 times 2. So 2pq equals 0.4663. Right? Rounded to 2 decimal places, so 0.46. Right? Okay. 122. A typical human lymphocyte has a radius of about 10 micrometers, while a typical bacteria has a radius of 1. Assuming that both are perfectly spherical, how many times larger is the surface area of a typical human lymphocyte compared to the surface area of a typical bacteria? I'm not going to do this one. I'm assuming it is easy enough, right? What is the volume, right? Um, yeah, calculating surface area of a sphere. What is that like? I don't know. They give you the, I don't even know the thing, but they should give it to you in the formula sheet, right? I believe. Yeah. Surface area of a sphere is 4 pi r squared, right? So just like that. Should be easy. If someone wants me to go over it, then I will, but otherwise, I hope you can do it. 123. One method for predicting the average temperature at which a short DNA molecule binds with this complement is to calculate a melting temperature using the following formula. Using the formula, what is the predicted TM for the short single-stranded DNA molecule shown above? Okay, right, should be easy, right? How many G's and C's, right? Just plug it in. I'm assuming that this can be done, right? Do I really have to count this? 1, 2, 3, 4, 5, 6, plus C, 1, 2, 3, 4, times 4, plus 2, A, 1, 2, 3, 4, plus T, 1, 2, 3, 4, 4. I don't know. I think that's right. 16 plus 40. 46. I feel like I didn't count this right because this C is not marked. Damn it. But yeah, you guys should be able to count the nucleotides probably better than I can, right? That's all you have to do. Calculate how many G's, how many C's, how many A's, how many T's, right? Just plug it in. Okay, 120. Okay, skip that. Oh, 124. As part of a study to observe the acidity of photosynthetic organisms in a temperate zone lake, measurements of light penetration were made at different water depths. The resulting results are presented in the graph below. Note that higher light penetration values indicate a greater amount of light reaching the water at that depth. According to the data above, what is the deepest water level at which light penetration is expected to be at least 200 micro, whatever that unit is. 200 is right here, right? So that's like right here, right? So what is that? 1.5 meters, right? I think anything between 1.2 and 1.8 is an acceptable answer, inclusive. Okay, an experiment on plant defenses was conducted on 3 identical groups of lima bean plants. The first group was treated with a solution containing a chemical similar to an organic compound that is released by herbivore damaged plants, solvent plus chemical. Second group was treated with only the solvent used in the first treatment. The third group was left untreated. Right? 1 week after the treatment phase, the number of carnivorous ants found residing on the plants were determined. Results are provided below. Based on the information, how many times more ants are found on plants exposed to the solvent plus chemical? Solvent plus chemical is group 1, right? Compared to plants exposed to solvent only, right? So what is that, like 35 and 90, right? I don't know, 90 divided by 35, 2.5 or 2.6, right? It's like 2.57, so I would say like 2.6, right? I mean, this is more than 35, it's like 37, whatever. Anything between 2.2 and 2.6 inclusive is okay. Okay, let's take a 3-minute break and we'll start 5:56 on this. Okay. Catechol. A natural substance found in plants reacts with oxygen to produce benzoquinone in water, as represented by the chemical equation above. Reaction is catalyzed in plants by the enzyme polyphenol oxidase. Accumulation of benzoquinone in plant tissues results in the gradual appearance of a brown color. A student observes that lemon juice pH 2 slows the browning of apple slices. Student claims that lemon juice slows the browning process by altering the activity of polyphenol oxidase. To test the claim, the student distributes equal amounts of a dilute catechol solution to 6 identical test tubes. pH of each solution was adjusted as shown in the table, and the initial absorbance was recorded. Equal amounts of polyphenol oxidase were added to each reaction tube. After 10 minutes at room temperature, the absorbance of 389 nanometer light was measured for each sample. Solutions containing greater concentrations of benzoquinone absorb more light. Changes in A389, absorbance of 389 nanometers, are shown in the table above below. Okay. So what do you see? Right, as your pH goes up, what happens to, or sorry, yeah, as your pH goes up, you see your absorbance go up to here, and then it goes back down, right? On the axis provided, construct an appropriately labeled bar graph to analyze the effect of pH on polyphenol oxidase activity, right? So literally, I'm assuming they have this, right? So just make an X. You have to label your axes, right? So probably X axis is going to be pH, right? And then your Y axis is going to be absorbance of 389 nanometers, right? Make sure to label, right? Probably I would, right? Again, also remember, x-axis is where your dependent variable goes, y-axis, independent variable, and then your dependent variable here, right? First one is 3 points, right? So part A is going to be 3 points, right? 1 point for your axes being labeled, 1 point for your axes being scaled, and then 1 point for data correctly plotted with your pH as your independent variable. So your pH has to be here, and your absorbance 389 will be here, right? So total 3 points. Part B. Based on the experimental results, estimate the optimal pH of the enzyme. Explain your estimate by connecting the experimental results to the structure and function of proteins. Okay, so what is your optimal pH? If you look at your change in absorbance, right? So it's going to be what? Probably 7, right? You have optimal absorbance at pH 7, right? 1 point, right? Um, you can also write that it's greater than 5 and less than 9, right? That is also an acceptable answer, but 1 point. Okay, your justification, right? What happens? How does pH affect your structure and function of protein, right? Um, pH can do multiple things, right? It can denature, right? It can change the structure of your active site, denature your protein, right? So if you have an optimal range, which does seem to be between 5 and 9, right? Anything outside of the optimal range will cause denaturation of your protein, which will change, right, its structure and therefore affect its function, right? So that as your explanation, 1 point, right? So 1 point for writing 7 or 5, between 5 and 9, 1 point for a valid explanation. Okay, so 3 points, 2 points, 3, 2. Make sure you're keeping track. Part C. Propose an appropriate control treatment for the experiment and describe how the control treatment would increase the validity of the results. Okay, so we have incubation, right, with our enzyme, right? Or change in absorbance with your enzyme. No enzyme, right, would be a control, right? How do you know that this is caused by your enzyme, right? Your control group would be no enzyme. That's it. Um, so it basically tells you if you do it with no enzyme, it tells you that the enzyme is necessary, right? Um, that is 1 point for the evidence, 1 point for the justification. Um, so total 2 points. Other possible answers. Reaction with denatured enzyme, right? And that's the same description as the other one, right? Whether you have no enzyme or denatured enzyme, and it confirms that your enzyme is necessary. Another control could be anaerobic conditions, no oxygen, right? Which confirms, if you look at that reaction, right, it confirms that oxygen is necessary, right? You can also do reaction without catechol, right? And that would confirm that catechol is necessary. So as long as you have any of those controls, that should be okay. Is there anything else? I don't know. Did you guys put something else and you want to ask? If you do at any time, like, is this okay? Just let me know. Okay, genetic engineering can be used to disrupt specific genes in the genome of an organism, predict how the browning of apple slices would be affected in a strain of apples that was genetically engineered to lack functional copies of the gene encoding polyphenol oxidase, right? Easy. It's not going to brown, or it will brown, or sorry, it will cause, this guy is going to catalyze this reaction, right? If you have no polyphenol oxidase, right, your apples will probably brown extremely slowly, right? Because benzoquinone is gonna take much longer to be produced, right? So will it still brown? Yes, eventually, right? But it would take a much, much longer time, assuming that you have no enzyme to catalyze that reaction. Okay, so 1 point for prediction, 1 point for justification, so total 2 points. Right? So total for your long questions, total of 10 points. Right? So mark your points out of 10. Okay. Number 2. Loeys-Dietz syndrome, type 2B, is a human genetic disorder associated with mutations in the LDS2B gene. Researchers have proposed that the LDS2B gene produces a growth factor receptor protein that regulates cell signaling pathways. The predicted product of the gene is a 565 amino acid protein with structural regions that bind to signaling molecules, span the plasma membrane, and have protein kinase activity. Protein kinases are cellular enzymes that transfer phosphate groups from ATP to target proteins. A representation of the LDS2B gene is shown in figure 1. The 7 exons, introns, the lines between the boxes, start and stop signals are represented. Portions of the gene that encode different structural regions of the proteins are shown above. Location of a methionine to valine substitution at position 425 is indicated, right? So we have a mutation here. Okay, based on a pedigree of a family in which one parent in the first generation has Loeys-Dietz syndrome, type 2B, a researcher claims that the disorder is an autosomal dominant condition. Describe 2 characteristics of the pedigree that would support the researcher's claim. So what would support the fact that this is a dominant disorder? Autosomal dominant, right? First of all, right, if it's autosomal, it means what? Males and females are affected equally, right? Because what do we know about sex linked? It's going to affect males at a greater proportion, right? And then how do we know if it's dominant, right? It means that if you have it in one generation, right, it will not disappear in the next generation, right? If you have a dominant gene, right, or dominant disorder in one generation, the next generation will also have it, right? It will not disappear. Yeah. It also means that there's no such thing as a carrier or a healthy carrier, right? Whether you're homozygous dominant, recessive, you'll also have it, right? Healthy individuals should be very rare, right? Okay. So one for autosomal, one for dominant, right? One for each explanation, 1.1 point. Part 2. Calculate the minimum number of nucleotides required in the coding region of the LDS2B mRNA molecule to produce and terminate the 565 amino acid polypeptide. Provide 2 reasons that the number of nucleotides in the mature mRNA may differ from the number of base pairs in the gene. Okay. You have 565 amino acids, right? 565 amino acids, right? How many nucleotides times 3? Right, so 565 times 3 is 1695, right? What else do you need though? Start codon plus 3, stop codon plus 3. What is that? Plus 6, 1701, right? And you need a rationale, right? Why is it 1701, right? Because each amino acid is coded by a total of 3 nucleotides, right? And then you need a start codon and you need a stop codon, right? So the minimum number that you would need is 1701 base pairs, right? So if you did this, you get 2 points. If you just wrote 1695 as your answer, this is 1 point. 1701 is 2 points. If you put 1698, you can also get 2 points. Right, but you need your rationale as well. Okay, so again, right, 2 reasons that the number of nucleotides in the mature mRNA may differ from the number of base pairs in the genes, right? Remember, first one, you have what? mRNA processing, right? So what do? First one, splicing, right? You remove your introns and you splice your exons together. So what is that going to do? That's going to change, right, how many nucleotides you have, right? You also add a 5' guanine cap and a 3' poly A tail, right? That's going to change the number of nucleotides that you have as well, right? Yeah, those would be 2, right? I don't know if there's anything else that you guys want to put and you want to know if it works. Let me know. Okay. Okay, part C. One genetic change associated with the disorder results in a methionine to valine substitution at amino acid position 425. Using the codon table, predict a DNA point mutation that will result in a methionine to valine substitution. So methionine is what? AG, right? Right here. And then you have valine, which is here, right? Probably what? This guy was turned, you have A to G, right? Because GUG codes for valine. Good? Okay. So Oh, sorry. I didn't give you guys the points for this, right? So this calculate part, 2 points. Provide 2 reasons, 2 points. Right? Predict a DNA point mutation. This is going to be 1 point. This first one is 2 points. And so we have 7 points so far. The last one, 3 for each feature, so 1 for each, right? Propose 3 features of a model to connect the genetic mutation you predicted with the activity of a signaling pathway involving the LDS2B gene product, right? Okay. So it happened in the protein kinase region, right? So what does that mean? Basically, this is where you are going to have your, like, I don't know what kind of receptor this is. Right? I don't know if this is like um, a GPCR. I don't think it is, right? But anyways, right? If this is your receptor, right, you have your binding region here, right, where your signal molecule is going to bind, right? That's coded by these guys, right? This one is your transmembrane region, so the part within your region, right? And then this dark area is where it's going to attach to a protein kinase, right? What is a protein kinase? Anything that's a kinase, these are activators, right? Um, Kinases phosphorylate things, right? Phosphorylate things means that you're taking a phosphate from ATP and adding it onto something else, right? And what does adding phosphate do? It activates it, right? So you have a mutation in your protein kinase region, right? So you can receive the signal, but within your cell, it might not do anything. Right? Um, so 3 features of a model, right? So we don't really know, right? Um, we don't really know what's going to happen, right? What is the genetic activity you predicted, right? So mutations, right? Produces a growth factor receptor protein that regulates cell signaling, blah, 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 bind to signaling, blah, 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 blah. Show representation of the, yeah, they don't really tell us anything, right? So you can do uh, loss of function, gain of function, anything that works. But the mutation will cause a structural change in your polypeptide, right? And it could activate signaling, right? So you can write that. Or you can write that it decreases your signaling pathway, right? It really depends which way you go. They don't give you any information on Lloyd's Dietz. So it doesn't matter if you say it's going to cause, right? Because remember, um, it could, yeah, it could do either one. You don't know. So if you're going to do loss of function, as long as your 3 points are related to that, that should be okay. If you're going to do gain of function, as long as it's along the lines of that, it should be okay, right? So 3 points maximum, right? As long as it's consistent. Right, so total 3 points. Okay. Are you guys all juniors? Junior? Are you senior? Junior? Fun. Okay, amino acid at position 104 of beta hemoglobin. Okay, beta hemoglobin is a highly conserved protein among vertebrates. The identity of amino acid 104, one of the variable amino acids in the polypeptide, in 4 vertebrate species is shown in the table above. A student constructed the phylogenetic tree shown in figure 1 by comparing the amino acid sequences of beta hemoglobin in the 4 species. The student placed lysine at an incorrect position on the phylogenetic tree uh, in an attempt to show when the mutation affecting amino acid 104 occurred, right? This is not correct. Refine the student's model by placing lysine at the correct position on the phylogenetic tree to indicate where the arginine to lysine mutation most likely occurred. Okay, so where did this occur? If you look at this, right, all of them have arginine except for the gorilla. So where should lysine be? It should be right here, right? Provide one piece of reasoning to support your placement of lysine on the phylogenetic tree, right? Again, right? So 1 point if you put it right here, and then 1 point for the reasoning. So what is the reasoning, right? Gorillas are the only ones that have that arginine to lysine um, mutation, right? Um, so it has to be occur only for the line or the branch leading to gorillas, right? So 1 point for the placement, refine, 1 point for provide, right? So it should be here. Okay, part B. Predict the most likely single mutation affecting amino acid 104 in the species and justify how beta hemoglobin can function normally in all 4 species despite the mutation. So what is likely? How do we go from arginine to lysine? So we have lysine a key, arginine a key, right? So probably, right, it's an A to G point mutation, right? Substitution, right? Um, right? But this is talking about RNA, right? Wait, we went from arginine to lysine, G to A, sorry, right? It's either a G to A point mutation. In the DNA, it would be what? A C to T mutation, right? You could put either one, right? But that is 1 point, right? Now, justification, right? As in, how come you still have fully functional beta hemoglobin even though you have this mutation, right? Because again, substitution doesn't cause frame shift, right? So even though it didn't cause a frame shift mutation, so it only affected one amino acid. It could just be that the structure wasn't significantly altered, therefore it still works, right? You can talk about how if arginine and lysine, there are amino acids with similar R groups, then your function can still be normal, right? Because, um, like for example, if arginine and lysine, I have no idea if it's true, but if they both have polar R groups, right, then when you fold the protein, right, the R group interaction will be not affected significantly, will not be affected significantly, right? So 1 point, 1 point, 1 point, 1 point. So total of 4 points. Okay, number 4. Bacteria and fungi are found in all ecosystems on Earth. A scientist claims that fungi are keystone species in a particular terrestrial ecosystem. To test this hypothesis, the scientist divides an area within the ecosystem into multiple small plots of similar size. Equal numbers of plots are then randomly assigned to one of 3 treatment groups. Plots in the first group are sprayed with a water-soluble substance that selectively kills fungi. Plots in the second group are sprayed with a water-soluble substance that selectively kills bacteria. Plots in the third group are sprayed with the same volume of water only. After a certain period of time, the scientist then measures the amount of accessible nutrients in the soil in each plot. Okay, um, independent variable, right? Dependent variable, independent variable, control, we have all of that, right? Um, your treatment, your water soluble, whatever, your uh, the substances that you sprayed, right? That's your independent variable, right? The amount of nutrients is your dependent variable. Yeah. Okay, part A. Describe one common role of both bacteria and fungi in the movement of energy and matter in ecosystems, right? What do bacteria and fungi have in common? Decomposition, right? So in ecosystems, right, they decompose matter, right, and play a role in nutrient cycling. Okay, part B. Describe how analyzing the data from the treatment group sprayed with water only will increase the validity of the experimental results, right? Water only, it proves a baseline, right, of how much nutrients are normally in the soil, right? Because if we measure, right, nutrients with group water soluble substance that selectively kills fungi, water soluble substance that selectively kills bacteria, right? And then we don't have a control, right? We don't know what normal is, right? What is our baseline, right? So yeah. Um, so any control, right? You need a control because if I remove bacteria and I compare it with if I don't have a control, how do I know that that change is due to the absence or lack of bacteria, right? How do I know that this result is due to the lack or absence of fungi, right? There's no way for us to tell if I have no control. Okay, part B. Describe how analyzing the data from the treatment. Oh, sorry. Part B is 1 point, right? Part A, 1 point. Part B, 1 point. Part C. Based on the scientist's claim, predict which treatment group will have the smallest amount of accessible nutrients in the soil at the end of the experiment. Justify your prediction in terms of the relative impact of the keystone species in the environment. Well, according to this guy, right, he claims that fungi are keystone species, right? So if you remove fungi, then what should happen? You would have the least amount of nutrients, right? So group 1, right? And that's 1 point, right? Why, right? Because what do we know about keystone species? Or what makes a keystone species a keystone species, right? Keystone species have what? Disproportionately large, right? Impact on their ecosystem, on the ecosystem, right? Um, like starfish are a keystone species. Beavers. Oh wait, beavers are foundation species. Um, yeah. Anyway, so 1 point, 1 point, total of 2 points. Right. So number 4, also total of 4 points. Okay, number 5. Tetrodotoxin is a neurotoxin that blocks the sodium channels of the neuronal membrane. The pufferfish, which contains TTX in its liver, is considered a delicacy in Japan, even though eating incorrectly prepared pufferfish can be fatal to humans. Describe 2 functions of neurotransmitters at the postsynaptic membrane. Okay. So I mean, what do they do? Neurotransmitters, they bind, right? At your postsynaptic membrane, what do? Bind to receptors, right? Um, what does binding to the receptors do? It can cause what? Initiation, right, or firing of an action potential. If you have enough neurotransmitters attaching, right, to your postsynaptic dendrites, right, then that can cause enough uh, if it reaches plus 40, right, or it can trigger another action potential in your postsynaptic membrane, right? That's how you get transmission of a signal from one neuron to another, right? So will it cause it to reach your threshold potential so that it initiates, right, depolarization or an action potential, right? Um, Let me see. Now, what's going to happen, right? So 2 points, right? Based on the information provided above, explain how TTX most likely interferes with the transmission of information across synapses, right? What happens if you block sodium channels? No action potential, right? Because remember, in your action potential looks like this, right? What happens here, right? If you get an external stimuli, right, your sodium channels leak. If it reaches threshold potential, right, all of your what should we call it? Sodium channels are going to swing open, right? Your voltage gated sodium channels. You get an influx of sodium, which causes depolarization, right? Now, if you block sodium channels, you cannot have depolarization, which means you cannot fire an action potential, which means that your brain cells can no longer communicate with each other, and you will die very, very quickly, right? Um, yeah. So, explained, right? Again, no action potentials because, again, um, your sodium, you cannot get a sodium influx into your axon, right? And therefore, right, um, if you have no signal, right, then what happens? You cannot transmit that signal to your axon terminal to open your, to initiate, right, release of neurotransmitters, right? Nothing happens, right? Okay, so that will be 1 point for what happens, and then 1 point for why, so 2 points. Right, so evident, let me just read the evidence, no action potential in neurons, reasoning, sodium cannot flow across neuronal membranes, right, you can do that, 1 point, 1 point. Another option is no reaction in your postsynaptic cell because calcium isn't released, right? Because remember, what does calcium do? When your action potential reaches, or sorry, when your action potential reaches your axon terminal, right? That causes influx of calcium ions, which is going to cause your vesicles containing neurotransmitters to move and release your neurotransmitters into the synapse. So that will not work either. So you can write that 1 point, 1 point. So total 4 points. Okay. In a study of bacterial communities, researchers investigated interactions among naturally occurring strains of Vibrio bacteria. The researchers found that some Vibrio bacteria secrete antimicrobial substances that inhibit the growth and reproduction of other bacteria, antagonism. The researchers also found that some strains of Vibrio bacteria produce no antimicrobial substances themselves, but instead live in close proximity to other bacteria that produce antimicrobial substances to which they are not susceptible. Cooperation. By testing approximately 35,000 pairs of naturally occurring strains of Vibrio bacteria, The researchers identified 830 antagonistic interactions among genetically related Vibrio strains. The graph represents the probability of antagonism between any two given strains of Vibrio bacteria as a function of genetic relatedness, genetic distance. The greater the genetic distance, the less related are any two bacterial strains. Part A, describe one type of molecular evidence that can be used to determine the genetic relatedness of bacterial strains isolated from naturally occurring populations. Okay, how do we see if something is related? Multiple ways, right? DNA, DNA sequencing, right? Comparison of DNA sequencing, um, amino acid sequencing, right? So we can take a protein of bacteria A, bacteria B, compare the amino acid sequences of the proteins, right? I don't know. That's probably the one. I mean, you can sequence anything, right? Different proteins, different genes, and compare them, which is probably the most common one, right? But more probable than just doing a full DNA sequence comparison, right? So our gene sequencing, protein sequencing, something like that. Right, 1 point. Part B, using the information presented in the graph, describe the relationship between antagonism and genetic relatedness in the communities of Vibrio bacteria sampled for the study. Using the information, describe the relationship between antagonism and genetic relatedness. Okay. So antagonism means what? You see that when your genetic distance increases, right, your probability of antagonism increases, right? So if the more similar you are, right, the less likely you are to have antagonism, right? So like antagonism is basically like, if you're not related to me, like I'm gonna basically release a substance that kills you, right? So that you stay away, right? But if you are related, right, then the likelihood of that goes really down, right? So the greater the distance, the greater the probability of antagonism, right? Direct relationship that we have, right? So that is 1 point, right? As long as you're able to explain that relationship, right? Um, you do see that, like, I don't know what that is, like 0.013-ish, 25, right? 0.12, right? That's when your probability of antagonism greatly increases, right? So your antagonism stays stable as long as you're you have a low genetic distance, right? Increases sharply as that distance increases. 1 point. Part C. Provide reasoning to show how the results of the study support the claim that cooperation among Vibrio bacteria can increase the fitness of some individual bacteria living in a natural environment. Okay, so what do you notice, right? Like I mentioned before, right? This bacteria, right? They keep unrelated species away from them, right? So they're keeping the levels of unrelated strains down, right? Um, and by removing all of those different strains, now you have more resources, right, available for strains that are more closely related, right? So essentially, you're reducing competition, right, which helps your chances of survival, right? That is 1 point. Okay, according to the chemiosmotic model proposed by Peter Mitchell in 1961, an electrochemical gradient is linked to the synthesis of ATP in mitochondria. Construct an explanation of the chemiosmotic model by doing each of the following. Make a claim about the role of the inner mitochondrial membrane in ATP synthesis. Okay, so um, what does the inner mitochondrial membrane do in ATP synthesis, right? Multiple things. First main thing is what? It creates, right, proton gradient, right? It creates a proton gradient that is going to be necessary for ATP synthesis, right? You could also say uh, your inner mitochondrial membrane is the location where, right, ATP synthesis occurs. You could also say um, electron transport chain is located in your inner mitochondrial membrane, right? So any of those works, right? Creates a proton gradient, maintains a proton gradient, establishes a proton gradient, whatever you write, right? 1 point. Present one piece of evidence that supports the role you proposed in part A, right? Um, okay, so how do we know that it creates your proton gradient, right? Protons, right, are not permeable through your membranes. It's pumped out, right, by your protein complexes 1, 3, and 4, right? Um, that are embedded as part of your electron transport chain, right? And your protons are pumped against your concentration gradient, right? Um, yeah. I don't know. Well, yeah. So anything along the lines of that, right? And then what happens, right? Your protons, now that you have a greater concentration of protons, right? What happens? The only way that they can flow back, right, to establish equilibrium is through ATP synthase, right? And the flow of protons down its concentration gradient is what drives ATP synthase to make ATP, right? 1.1 point, right? So anything basically explaining how your inner mitochondrial membrane is where your electron transport chain is located, and as long as you can explain the role of your inner mitochondrial membrane in terms of your electron transport chain, ATP synthase, that should give you 1.1.3 points. Right, so if you want to say like, why does this happen and why does that happen? It's like weird. Right, so a total of 3 points. Okay, in 1952, Alfred Hershey and Martha Chase used radioactive phosphorus and radioactive sulfur to selectively label the DNA and proteins of bacteriophage T2, a virus that infects bacteria. After incubating the labeled bacteriophage particles with E. coli and separating extracellular phage particles from the bacteria, Hershey and Chase measured the amounts of radioactive phosphorus and sulfur inside affected E. coli cells and in the liquid growth medium outside the E. coli cells. Okay, so part A. Predict the experimental result that would best support the claim that DNA is a source of heritable information and provide reasoning to explain how the result supports the claim. Okay, so this was the classic experiment, right? So you had two groups of a virus. One, you grew it in a solution of radioactive phosphorus, one in radioactive sulfur. Why? Phosphorus is only in DNA, sulfur is only in proteins, right? And how do bacteriophages work? When they infect a bacteria like E. coli, right? They inject their DNA, or sorry, they inject their genetic material into the host cell, right? So if the genetic material is DNA, right, then when you grow your T2 phage in phosphorus, right, you're going to see radioactivity transferred from your virus to your E. coli, right? If protein is your genetic material, then when you grow your phage, right, in radioactive sulfur, you're going to see radioactivity transfer from E. coli to your bacteria, right? And that would prove, right, which one is the genetic information, right? Obviously, we know that DNA is our genetic information. This is the classic experiment that finally confirmed it, right? Okay. Yeah. So how do we, what was the result, right? That DNA is a source of heritable information, right? The majority of, uh, you only found uh, radioactivity in your E. coli, right? When it was grown in radioactive phosphorus, right? That's it, right? Remember when you separate it, right? You form a pellet. You're gonna centrifuge it. You have your pellet and then your supernatant, right? Your supernatant is gonna contain your virus particles, right? Why? Virus particles are way smaller than bacteria, right? In your pellet, what do you have? You're going to have your E. coli, right? So if you find the majority of your radioactivity in your supernatant, not in your pellet, right, that tells you that that is not the genetic information, right? So when you grew it in radioactive phosphorus, the majority of your radioactivity was found in the pellet, meaning that it transferred from your virus to E. coli, right? So that is the answer. 1 point for prediction, 1 point for the explanation, right? So you should have picked radioactive phosphor, right? And then be able to explain it. Total 2 points. Okay. Um, Part B. Bacteriophages, like other viruses, consist primarily of a protein coat and packaged DNA. Describe the function of one critical enzyme in bacterial cells that is necessary for replicating bacteriophage DNA. Okay, so what do we need to make DNA? DNA polymerase, right? DNA polymerase is necessary in order to replicate any DNA, right? I mean, you could talk. They say you can talk other. You can have other ones, helicase, topoisomerase, ligase, right? But really, the main enzyme is going to be DNA polymerase. Yeah, 1 point. Total 3 points. 1, 1, 1. Yeah, we finished early. Count it up. Count up your scores. Let me. Do this post it on Zoom? Well, yeah, but I mean for online people. I do not know. Would you like to but for the FRQs too? Oh really? Okay. Yes. Okay. Yeah. You guys have any questions? Story time? No. Just make sure you guys have your scores so that you guys can give it to the nice person up front. Um, no questions? Concerns? Are you guys taking it all in person this year?
Yeah.
Sad. No COVID advantage. um, okay, then yeah. Well, by the way, when is the biotech? I know chem is the first day. When is it? Do you know? May 3rd? That's the first week too, huh? Okay, well, you guys are free to go. We finished going over the test early. Unless anyone has questions, or you guys want to review some topic. I mean, it's spring break. You guys should go, like, sleep. On vacation, right? No, you're a junior. You can't go on vacation. Oh. Well, you lost your only opportunity. You're done. No more vacation for you. Study for your test. Then why go on vacation? That's even more depressing. Like think about like you're in like Mexico, right? Everyone's having fun. You're sitting at the beach with your laptop like studying. Really? You'd want to do that? Because I know what would happen to me. I would not study at all. I would plan on studying, take everything, and then not do anything. And then I'll come back and feel like shit. I'll be hating myself like, why didn't I do anything? I mean, that is also an option. Or just study at home. It's depressing either way, don't you think? No? You feel fun by studying in a new location. That is sad that that's your idea of fun. Bye.